हेलो फ्रेंड्स, माय नेम इज सिद्धिकांत सिक्वल और मैं आप सभी का स्वागत करता हूँ हमारा अपना प्यारा सा चैनल ए एन पी बाय एस जे। तो चलिएगा शुरू करते हैं आज बायोमोलिक्यूल्स एनसीईआरटी लीडिंग लाइन बाय लाइन। There is a wide diversity in a living organism in our biosphere. Now, question that arises in our minds is, are all living organisms made up of the same chemicals? That is, elements and compounds. You have learned in a chemistry how elemental analysis is performed. If we if we perform such an analysis on a plant tissue, animal tissue, or a microbial paste, we obtain a list of elements like carbon, hydrogen, oxygen, and several others and their respective content per unit mass of a living tissue. If the same analysis is performed on a piece of a Earth's crust as an example of a non-living metal, we obtain a similar list. What are the differences between the two lists? In absolute term, no such difference could be made out. All the elements present in a sample of Earth's crust are also present in a sample of a living tissue. However, the closer examination reveals that the sorry, the relative abundance of carbon and hydrogen with the respect to other elements is higher in any living organisms than in the Earth's crust. 9.1. How to analyze a chemical composition? How to analyze a chemical composition? We can continue asking in the same way what type of organic compounds are found in a living organisms. How does the how does one go about finding the answers? To get the answer, one has to perform a chemical analysis. We can take any living tissue, a vegetable or a piece of a liver, etc., and grind it in a trichloroacetic acid CL3CCOOH using a mortar and a pestle. We obtain a thick slurry. If we were to stain this through cheesecloth or cotton, we would obtain two fractions. One is called as a filtrate or a more technically the acid soluble pool, and second is a latentate or the acid insoluble fraction. Scientists have found thousands of organic compounds in the acid soluble pool. In higher classes, you will learn about how to analyze a living tissue sample and identify a particular organic compounds. It will suffice to say here that one extracts the compounds, then the subject the extract to various separations techniques till has separated a compound from all other compounds. In other words, one isolates and purifies a compound. Analytical techniques, when applied to the compound, give us an idea of the molecular formula and the pro- probable structure of the compound. All the carbon compounds that we get from a living tissue can be called as biomolecules. However, living organisms have also got inorganic elements and compounds in them. How do we know this? A slightly different but destructive experiment has to be done. One one weighs a small amount of a living tissue, say a leaf or a liver. This is called wet and dried. All the water evaporates. The remaining materials gives dry weight. Now, if the tissue is fully burnt, all the carbon compounds are oxidized to gaseous form, that is CO2 water vapor and removed. What is remaining is called ash. This ash contains inorganic elements like calcium, magnesium, etc. Inorganic compounds like sulfate, phosphate, etc. are also seen in the acid soluble fraction. Therefore, elemental analysis gives elemental composition of a living tissue in the form of hydrogen, oxygen, chlorine, carbon, etc. While analysis is for compounds gives an idea of the kind of organic and inorganic constituents present in a living tissue. From the chemistry point of view, one can identify fractional groups like aldehyde, ketones, aromatic compounds, etc. But from a biological point of view, we shall classify them into amino acid, nucleotide, bases, and fatty acids, etc. Amino acids are organic compounds containing an amino group and an acidic group as a substituent on the same carbon. That is the alpha carbon. Hence, they are called alpha amino acids. They are substituted methanes. There are four substituted groups occupying the four valency positions. These are hydrogen, carboxylic group, amino group, and a variable group denoted as a R group. Based on nature of a R group, there are many amino acids. However, those which occur in a proteins are only of 20 types. The R group in the R group in this proteinaceous amino acids could be hydrogen. The amino acid is called glycine. A methyl group, alanine, hydroxymethyl, serine, etc. Three of the 20 are shown in figure 9.1. The chemical and the physical properties of amino acids are essentially of the amino and carboxyl and are functional groups. Based on the number of amino and carboxyl groups, they are acidic. Example, glut- glutamic acid, basic lysine and neutral valine amino acids. Similarly, there are aromatic amino acids, that is glycine, phenylalanine, and tryptophan. A particular property of amino acids in the ionizable nature of NH2 and COOH groups, hence insoluble of a different pH. The structure of amino acid changes. Lipids are generally water insoluble. They could be simple fatty acids. A fatty acid has a carboxylic group attached to an R group. The R group could be a methyl CH3 or ethyl C2H5 or a higher number of CH2 groups, that is a 1 carbon to 19th carbon. For example, palmitic acid has a 16 carbon including carboxyl carbon. Alkydonic acid has a 20 carbon atoms including the carboxyl carbon. Fatty acids could be saturated without double bond or unsaturated with one or more carbon carbon double bonds. Another simple Another simple lipid is glycerol, which is a trihydroxypropane. Many lipids have both glycerol and fatty acids. Here, the fatty acids are found esterified with glycerol. They can be then monoglycerides, diglycerides, and triglycerides. These are also called fats and oils based on melting point. Oils have a lower melting point, example gingerly oil, and hence remains as oils in a winter. Can you identify a fat from the market? Some some lipids have a phosphorus and a phosphorily, phosphorily, phosphorylated organic compounds in them. These are phospholipids. They are found in a cell membrane. Lactic Lectin is a one example. Some tissues, especially the neural tissue, have lipids with more complex structures. Living organisms have a number of carbon compounds in which heterocyclic links can be found. Some of these are nitrogen bases adenine, guanine, cytosine, uracil, and thymine. When found attached to the sugar, they are called nucleoside. If the phosphate loop is also found esterified to the sugar, they are called nucleotide. Adenine, ado, adenosine, guanosine, thymidine, uridine, and cytidine are nucleosides. Adenylic acid, thymidyllic acid, guanylylic acid, uridyllic acid, and cytidyllic acid are nucleotides. Nucleic acids like DNA and RNA consists of nucleotides. Only DNA and RNA functions as a genetic material. 19.2 Primary and secondary metabolites. The most exciting aspects of chemistry deals with isolating thousands of compounds, small and big, from living organisms, determining their structures and, if possible, synthesizing them. If one were to make a list of biomolecules, such a list would have thousands of organic compounds, including amino acids, sugar, etc. For reasons that are given in the section 9.10, we can solve we can call these biomolecules as a metabolites. In animal tissue, one notices the presence of all such categories of a compound shown in figure 9.1. These are called primary metabolites. However, when one analyzes plant, fungal, and microbial cells, one would see thousands of compounds other than these called primary metabolites. Example: alkaloids, flavonoids, rubber, essential oils, antibiotics, color pigment, scents, gums, species. These are called secondary metabolites. While primary metabolites have identified functions and play known roles in a normal physiological process, we do not at the moment understand the role of functions of all the secondary metabolites in the host organisms. However, many of them are useful to human welfare. Example: rubber, drugs, species, scents, etc. Lubricants, pigments. Some secondary metabolites are ecological important. In the later chapter and years, you will learn more about this. 9.3 Biomacromolecules. There are one feature common to all those compounds found in the acid soluble pool. They have molecular weights lagging from 18 to around 800 18 to 800 Dalton, that is the Da approximately. The acid insoluble fractions has only four types of organic compounds, that is protein, nucleic acids, polysaccharides, and lipids. These classes of compounds, with the exceptions of lipids, have a molecular weight in the range of 10,000 Dalton and above. For this very reason, biomolecules, that is chemical groups found in a living organisms, are of two types. One, those who have a molecular weight less than 1000 Dalton and are usually referred to as micromolecules or simply biomolecules, while those which are found in the acid insoluble fractions are called macromolecules or biomacromolecules. The molecules in the insoluble fractions, with the exceptions of a lipid, are polymeric substances. Then why do lipids, whose molecular weights do not exceed 800 Dalton, come under the insoluble fraction, that is macromolecular fractions? Lipids are indeed small molecular weight compounds and are present not only as such compound and are present not only as such but also arranged into the structures like cell membrane and other membranes. When grind a tissue, we are we are disrupting the cell structure. Cell membrane and the other membranes are broken into pieces and form a vesicles which are not water soluble. Therefore, these membranes fragments in the form of vesicles get separated along with the acid insoluble pool, and hence in the macromolecular fraction. Lipids are not strictly macromolecules. The acid soluble pool represents roughly the cytoplasmic composition. Sorry, the macromolecules form the macromolecules from cytoplasm and organelle becomes the acid insoluble fractions. Together, they represent the entire chemical composition of a living tissues or organisms. In summary, if we represent the chemical compositions of a living tissue from abundance point of view and arrange them class-wise, we observe that water is the most abundant chemicals in the living organisms. Components and their percentage of a total cellular mass: water 70 to 90%, protein 10 to 15%, carbohydrate 3%, lipid 2%, nucleic acid 5 to 7%, and ions 1%. 9.4. Proteins. Proteins are polypeptide. They are linear chains of amino acids linked by the peptide bonds as shown in figure. Each protein is a polymer of amino acids, as there are 20 types of amino acids. Example: alanine, cytidine, proline, tryptophan, lysine, etc. A protein is a heteropolymer and not a homopolymer. A homopolymer has only one type of a monomer repeating n number of times. This information about the amino acid content is important as a later in your nutrient lessons. You will learn that the certain amino acids are essential for our health and they have to be supplied through our diet. Hence, dietary proteins are the source of essential amino acids. Therefore, amino acids can be essential or non-essential. The latter are those which our body can make. While we get essential amino acids through our diet or food, proteins carry out many functions in living organisms. Some transport nutrients across cell membrane, some fight infectious organisms, some are hormones, some are enzymes, etc. The collagen is the most abundant protein in animal world and ribulose biphosphate carboxylase oxygenase, that is rubisco, is the most abundant protein in the whole of the biosphere. Then some proteins and their functions. Collagen intercellular ground substance, trypsin function enzyme, insulin function hormone, antibody function fight infectious agents, receptors function sensory receptions, smell, taste hormone, GLUT 4 functions enables glucose transport into cell. So yeah, 9.5 polysaccharides. The acid insoluble pellets also has a polysaccharides. Carbohydrates has another class of macromolecules. Polysaccharides are a long chains of sugars. They are threads, literally a cotton thread containing a different monosaccharides as a building blocks. For example, cellulose is a polymeric polysaccharide consisting of only one type of monosaccharide, that is glucose. Cellulose is a homopolymer. Starch is a variant of this but present as a storehouse of energy in plant tissue. Animals have another variant called glycogen. Insulin is a polymer of a fructose in a polysaccharide chain, say glycogen. The right end is called the reducing end and the left end is called as non-reducing end. It is the branches as shown in the form of the cartoon. Starch forms a helical secondary structures. In fact, starch can hold I2 molecules in the helical portion. The starch I2 is blue in color. Cellulose does not contain complex helixes, hence cannot hold I2. Plant cells where plant cell wall are made of cellulose. Paper made from plant pulp and cotton fibers is a cellulosic. There are more complex polysaccharides in nature. They have as building blocks amino sugars and chemically modified sugars, that is a glucosamine, n acetyl gluco glucosamine, etc. Exoskeletons of arthropods, for example, have a complex polysaccharide called chitin. These complex polysaccharides are mostly homopolymers. 9.6 Nucleic acid. The other types of macromolecules that one would find in the acid insoluble fractions of any living tissue is the nucleic acid. These are polynucleotides. Together with the polysaccharides and polypeptides, these comprises the true macromolecular fractions of any living tissue or a cell. For nucleic acid, the building blocks is a nucleotide. The nucleotide has a three chemically distinct components. One is a heterocyclic compound, the second is the monosaccharide, and the third is the phosphoric acid or a phosphate. As you notice in figure 9.1, the heterocyclic compounds in the nucleic acids are the nitrogenous bases named as adenine, guanine, uracil, cytosine, and thymine. Adenine and guanine are the substituted pyruvines, while the rest of the substituted as pyrimidines. The skeletal heterocyclic links is called as a purine and pyrimidine respectively. The sugars found in the polynucleotides is either ribose, that is monosaccharide pentose, or a 2-deoxyribose. Nucleic acids containing a deoxyribose is called deoxyribose nucleic acid, that is RNA, while which can contain ribose is called ribose nucleic acid, that is RNA. 9.7 Structure of proteins. Protein, as mentioned earlier, the heteropolymers containing strings of amino acids. Structures of molecules means different things in a different context. In inorganic chemistry, the structure invariably refers to the molecular formulae, example NaCl, MgCl2, etc. Organic chemicals always write in two-dimensional view of the molecules while representing the structure of molecules, example benzene, naphthalene, etc. As a physics, sixth consecutive as a physics constitutes up the three-dimensional views of a molecular structures, while biologists describe the position structure of a four levels, the sequence of amino acids, that is potential positional information in the protein, which is first amino acid, which is a second, and so on, is called as primary structure. Primary structure of a protein. Protein is imagined as a line. The left end is represented by the first amino acid and the right end represented by the last amino acids. The first amino acid is also called as a N-terminal amino acid, and the last amino acid is also called as C-terminal amino acid. The protein threads does not exist throughout as an extended rigid rod. The thread is folded in the form of helix, that is similar to the revolving staircase. Of course, only some portions of the protein threads are arranged in the form of a helix. In proteins, only right-handed helix are observed. Other regions of the protein's thread are folded into other forms in what is called the secondary structures. In addition, the long protein chain is also folded upon itself like a hollow woolen ball, giving rise to the tertiary structure. This gives this gives us a three-dimensional view of a protein. Tertiary structures is absolutely necessary for the many biological activities of a proteins. Some proteins are some proteins are an assembly of more than one polypeptide or subunits. The manner in which these individuals folded polypeptides or subunits are arranged with respect to each other. Example: linear string of spheres arranged one upon each other in the form of cubes or plates, etc. is the Arctic is the architecture of a position, otherwise called as the quaternary structure of a protein. Adult human hemoglobin consists of four subunits. Two of these are identical to each other. Hence, two subunits of a alpha type and two subunits of a beta type, together constituting the human hemoglobin. 9.8 Enzymes. Almost all enzymes are proteins. There are some nucleic acids that behave like enzymes. These are called lipozymes. One can depict an enzyme by a line diagram. An enzyme, like any protein, has a primary structure, that is amino acid sequence of the protein. An enzyme, like any protein, has the secondary and tertiary structure also. When you look at a tertiary structure, you would notice that the backbone of the protein chain folds upon itself. The chain crisscrosses itself, and hence many crevices or pockets are made. One such a pocket is the active site. An active site of an enzyme is a cervix or a pocket into which the substrate fits. Thousands of thus the enzyme through their active sites catalyzes reaction at a higher rate. Enzyme catalysts differ from inorganic catalysts in many ways. But one major difference needs mention. Inorganic catalysts work efficiently at high temperatures and high pressure, while enzymes gets damaged at a higher temperature, say about 40 degrees Celsius. However, enzymes isolated from organisms who normally live under extremely high temperature. Example: hot vents and sulfur springs are stable and retain their catalytic power even at higher temperatures, up to 80 to 90 degrees Celsius. Thermal stability is thus an important quality of such enzyme isolated from thermophilous organisms. 9.8.1 Chemical reactions. How do we understand these enzymes? Let us first understand the chemical reactions. Chemical compounds undergo two types of changes. A physical change simply refers to the change in a shape without breaking the bonds. This is a physical process. Another physical process Another physical process is a change in a state of matter. When ice melts into water, or when water becomes a vapor, these are also physical processes. However, when bonds are broken and new bonds are formed during the transformation, this will be called chemical reactions. Example: Ba in bracket OH2 plus H2SO4, that gives BaSO4 plus 2H2O is in is an organic chemical reaction. Similarly, hydrolysis of a starch into glucose is an organic chemical reaction. A rate of physical or a chemical processes refers to a amount of a product formed per unit. It can be expressed as rate equals low P by low T. Rate can be also called a velocity if the direction is specified. Rates of physical and chemical processes are influenced by a temperature among other factors. A general rule is a thumb is that rate doubles or doubles or decreases by half for every 10 degree Celsius change in either direction. Catalyzed reactions proceed at a rate vastly higher than that of uncatalyzed ones. When enzyme catalyzed reactions are observed, the rate would be vastly higher than the same but uncatalyzed reactions, for example CO2 carbon dioxide plus H2O water. In the presence of a carbonic anhydrase H2CO, it gives H2CO3, that is carbonic acid, and vice versa also. In the absence of enzyme, these reactions is very slow, with about 200 molecules of H2CO3 formed formed in an hour. However, by using the enzyme present within the cytoplasm called carbonic anhydrase, the reaction speeds dramatically with about 6 lakhs molecules being formed every second. The enzyme the enzyme has accelerate the reactions rate by about 10 million times. The power of an enzyme is incredible indeed. There are there are thousand types of enzymes, each catalyzing a unit chemical or a metabolic reactions. A multiple steps chemical reactions when each of the step is catalyzed by the same enzyme complex or different enzymes is called a metabolic pathway. For example, glucose gives 2 pyruvic acid, that is C6H12O6 plus O2 gives 2C3H4O3 plus 2H2O is actually a metabolic pathway in which glucose becomes pyruvic acid through 10 different enzymes catalyzed metabolic reactions. So when you study respiration in chapter 12, you will study these reactions. At this stage, you should know that these very metabolic pathways with one or two additional reactions gives rise to a variety of metabolic end products. In our skeletal muscles, under the anaerobic conditions, lactic acid is formed. Under a normal and aerobic conditions, pyruvic acid is formed. In yeast, during the fermentation, the same pathway leads to the production of ethanol, that is alcohol. Hence, in different conditions, different products are possible. 9.8.2. How do enzymes bring about such high rates of chemical conversions? To understand this, we should study enzyme little more. We have already understood the idea of an active site. The chemical or the metabolic conversions refers to the reaction. The chemicals which is converted into a product is called substrate. Hence, enzyme, that is a protein with a three-dimensional structures including an active site, converts a substrate, that is denoted by S, into a product denoted by P. Symbolically, this can be easily depicted as S gives. It is now understood that the substrate S has to bind the enzyme at its active site within a given cleft or pocket. The substrate has to diffuse towards the active site. There is thus an obligatory formation of an ES complex, that is E stands for enzyme and S stands for substrate. This complex formation is a transient phenomenon during the state where substrate is bound to the enzyme active site. A new structure of the substrate called a transition state structure is formed very soon after expected bound expected bond breaking or a making is completed, the product is released from the active site. In other words, structure of substrate gets transformed into the structure of products. The primary the the pathway of this transformation go through the so must do must go through so-called transition state structure. There, there could be many more altered structural states between the stable substrate and the product. Implicit in this statement is the fact that all other intermediate structural states are unstable. Stability is something related to energy status of the molecule or the structure. Hence, when we look at this pictorially through the graph, it looks like a something as in figure 9.4. The y-axis represents the potential energy content and the x-axis represents the progression of the structural transformation or the states through the transition state. You would notice two things: the energy level difference between S and P. If P is at a lower level than S, then the S reaction is an exothermic reaction. One need not supply energy by heating in order to form the product. However, whether it is an exothermic or a spontaneous reaction or an endothermic or energy requiring reactions, the substrate yes has to go through go through a much higher energy state or transition state. The difference the difference in the average energy content of a yes from that of this transition state is called as activation energy. Enzyme eventually bring down this energy barrier, making the transition of a S to P more easy. 9.8.3 Nature of enzyme action. Each enzyme E has a substrate S binding site in its molecule, so that is a highly active reactive enzyme substrate complex ES is produced. This complex is short-lived and dissociates into the products P and the unchanged enzyme with other intermediate formation of the enzyme product complex EP. The formation of the ES complex is essential for catalysts. E plus ES, that gives EP, that gives E plus P. The catalyst cycle of an enzyme action can be described in the following steps. First, the substrate binds to the active site of the enzyme, fitting into the active site. The binding of the substrate induces the enzyme to alter its shape, fitting more tightly around the substrate. The active site of the enzyme, now in close proximity of the substrate, breaks the chemical bonds of the substrate and the new enzyme product complex is formed. 4. The enzyme released of the products of the reaction and the free enzyme is ready to bind to another molecules of the substrate and run through the catalytic cycle once again. 9.8.4 Factors affecting enzyme activity. The activity of an enzyme can be affected by a change in the condition which can alter the tertiary structures of the protein. These include temperature, pH, change in substrate concentration, or binding of a specific chemicals that regulate its activity. Temperature and pH. Enzymes are generally function in a narrow range of a temperature and pH. Each enzyme shows its highest activity at a particular temperature and pH, called as optimum temperature and optimum pH. Activity declines both below and the above optimum values. Low temperature preserves the preserves the enzyme in the temporarily inactive state, whereas a high temperature destroys enzyme activity. Because proteins are denaturated by high concentration of substrate. With the increase in the substrate concentration, the velocity of the enzymatic reactions rises as first. The reactions ultimately reaches a maximum velocity, that is V maximum, which is not exceeded by an further rise in concentration of the substrate. This is because the enzyme molecule are fewer than the substrate molecules, and after the saturation of these molecules, there are no free enzyme molecules to bind with the additional substrate molecules. The activity of an enzyme is also sensitive to the presence of a specific chemicals that bind to the enzyme. When the binding of the chemical suits of enzyme activity, the process is called as inhibition and the chemical is called as inhibitor. When the inhibitor is closely resembles the substrate in its molecular structure and inhibits the activity of the enzyme, it is known as a competitive inhibitor. Due to its close structural similarity with the substrate, the inhibitor competes with the substrate for the substrate binding site of the enzyme. Consequently, the substrate cannot bind, and as a result, the enzyme action declines. Example, inhibition of succinate dehydrogenase by malonate, which closely resembles the substrate succinate in the structure. Such competitive inhibitors are often used in the control of a bacterial pathogens. 9.8.5 Classification and nomenclature of enzymes. Thousands of enzymes have been discovered, isolated, and studied. Most of the enzymes have been classified into different groups based on the types of reactions they catalyze. Enzymes are divided into 6 classes, each with 4 to 13 subclasses and named accordingly by 4-digit number. Oxido, oxido, oxidoreductase or dehydrogenase. Enzyme which catalyze oxidoreduction between two substrates S and S. Example, S reduced X plus S oxidized, that gives S reduced plus S dash oxidized, that gives S oxidized plus S dash reduced. Transferase. Enzyme catalyze enzyme catalyzing a transfer of a group G other than hydrogen between a pair of a substrate S and S dash. Example, S minus G plus S dash gives S plus S dash minus G. Hydrolysis. Enzymes catalyzing hydrolysis of ester, ether, peptide glycosidic, C single bond C single bond halide or P single bond N bonds. Lysis. Enzymes that catalyze removal of a group from substrate by mechanism other than hydrolysis, leaving double bonds. Example, XY X C का साथ X C के X कार्बन के साथ Y कार्बन के साथ gives X minus XY plus C double bond C. Isomerase included all enzymes catalyzing interconversions of optimal geometric or positional isomers. Ligases enzymes catalyzing the linking together of two compounds. Example, enzyme which catalyzes joining of a CO, CS, CN, PO, etc. bonds. 9.8.6 Cofactors. Enzymes are composed of one or a several polypeptide chains. However, there are a number of cases in which non-protein constituents called a cofactor are bound to the enzyme to make the enzyme catalytically active. catalytically active. In these instances, in these instances, the protein portion of the enzyme is called apoenzyme. Three kinds of cofactors may be identified: prosthetic group, coenzyme, and metal ions. Prosthetic group are organic compounds and are distinguished from other cofactors in that they are tightly bound to the apoenzyme. For example, in peroxidase and catalysis, which catalyze the breakdown of hydrogen peroxide to water and oxygen. Heme is the prosthetic group and it is the part of the active site of apoenzyme. Coenzymes are also organic compounds, but their association with the apoenzyme is only transient, usually occurring during the courses course of a catalysis. Furthermore, coenzyme serves as a cofactor in a number of different enzyme catalyzed reactions. The essential chemical components of many coenzymes are vitamins. Example: coenzymes niclotamide, adenine, dinucleotide NAD and NADP contains the vitamins nicotines. A number of enzymes require a metal ions for their activity, which form coordination bonds with a side chains at the active site and at the same time form one or more coordination bonds with the substrate. Example: zinc is a cofactor of the proteolytic enzyme carboxypeptidase. Catalytic activity is lost when the cofactor is removed from the enzyme, which testifies that they play a crucial role in the catalytic activity of the enzyme. Summary: although there is a building diversity of living organisms, their chemical compositions of metabolic reactions appear to be remarkably similar. The elemental composition of a living tissue and a non-living matter appear also to be similar when analyzed qualitatively. However, a closer examination reveals that the relative abundance of carbon, hydrogen, oxygen is higher in living systems when compared to inanimate matter. The most abundant chemical in living organism is water. There are thousands of small molecular weight, that is less than 1000 Dalton biomolecules, amino acids, monosaccharides and disaccharides, sugars, fatty acids, glycerol, nucleotides, nucleosides, and nitrogen bases are some of the organic compounds seen in living organisms. There are 20 types of amino acids and 5 types of nucleotides. Fats and oils are glycerides, in which fatty acids are esterified to glycerol. Phospholipids contain, in addition, a phosphorylated nitrogenous compounds. Only 3 types of macromolecules, that is protein, nucleic acid, and polysaccharides, are found in living system. Lipid, because of their association with the membrane, separate in the macromolecular friction. Biomacromolecules are polymers. They are made up of a building of blocks are different. Proteins are heteropolymers made up of amino acids. Nucleic acids, RNA and DNA are composed of nucleotides. Biomacromolecules have a hierarchy of structures: primary, secondary, tertiary, and quaternary. Nucleic acids serve as a genetic material. Polysaccharides are components of a cell wall in plants, fungi, and also of the exoskeleton of arthropods. They also are storage forms of energy, example starch and glycogen. Proteins serve a variety of cellular functions. Many of them are enzymes, some are antibodies. Collagen is the most abundant protein in animal world and Libulose biophosphate carboxylase oxygenase Lubisco is the most abundant protein in the whole of the biosphere. Enzymes are the protein which catalyze a biochemical reactions in the cell. Lipozymes are nucleic acid with catalytic power. Proteinaceous enzymes exhibit a substrate specificity, requires optimum temperature and pH maximum activity. They are denatured at high temperatures. Enzymes lower activation energy of reactions and enhance greatly the rate of reactions. Nucleic acids carry hereditary information and are passed on from the parental generation to progeny. सो यहाँ पर अपन ने बायोमोलिक्यूल्स भी कंप्लीट कर लिया है और अपन ने ऑलरेडी पूरी ह्यूमन फिजियोलॉजी के पूरे 6 चैप्टर और उसके बाद ह्यूमन फिजियोलॉजी के 6 चैप्टर और स्ट्रक्चर ऑर्गेनाइजेशन इन एनिमल्स भी कंप्लीट कर लिया है। आज भी बायोमोलिक्यूल्स कर लिया है। तो जिन्होंने भी बायोमोलिक्यूल्स के पहले के लेक्चर नहीं देखे वीडियो, उनको इसी वीडियो के डिस्क्रिप्शन बॉक्स में मैं सबकी ये दे दूंगा लिंक, देख लेना। और कैसा लगा प्लीज़ कमेंट में बताओ। जब आप कमेंट में बताओगे तो मुझे पता चलेगा कि ये जो मैं वीडियो बना रहा हूँ ये आपके लिए यूज़फुल भी हो रहे हैं या नहीं, या मैं ऐसे ही बनाते जा रहा हूँ सिर्फ। तो प्लीज़ कमेंट में बताना। और सभी तो मेरी तरफ से हैप्पी न्यू ईयर। और मैं यहाँ इसके डिस्क्रिप्शन बॉक्स में ही मेरे टेलीग्राम चैनल का लिंक भी दे दूंगा, उसको ज्वाइन कर लेना। वहाँ पर मैं क्वेश्चन प्रैक्टिस स्टार्ट कर दिया हूँ और सबसे हाई वेटेज वाली चैप्टर से स्टार्ट किया हूँ, दैट इज़ मॉलिक्यूलर बेसिस ऑफ इनहेरिटेंस। और उसके बाद चैप्टर वाइज वेटेज वगैरह भी डालते जा रहा हूँ। तो प्लीज़ उसको भी ज्वाइन कर लेना और प्लीज़ मुझे कमेंट बॉक्स में बताना कि वीडियो कैसा लगा। सो थैंक यू फॉर वॉचिंग दिस वीडियो।